In April 2020, a remotely operated vehicle descended more than 600 meters into the Ningaloo Canyon off the coast of Western Australia. The lights swept across empty blackness, catching only marine snow drifting like dust. Then something curled into view, a thread of tissue coiled in a vast spiral, ring after ring, its outer edge stretching further than the camera could frame. The scientists aboard the research vessel Falkor began to measure. The outer coil alone spanned roughly 47 meters in circumference. The creature itself was estimated at close to 40 meters in total length. That number lands with a shock. A blue whale, the largest animal known to have ever lived, reaches around 30 meters at its very longest. This drifting spiral in the darkness was longer than any blue whale ever recorded. For a moment, it seems impossible. Nothing in the ocean should outmeasure the great whales. And yet here it hung, silent and glowing faintly, longer than a Boeing 747, longer than 3 school buses parked end to end. But the true shock is not the length. It is the twist hidden inside it. This record-breaking giant is not one animal. It has no single heart, no single mouth, no single body in the way a whale or a fish has a body. What the camera captured was not an individual creature at all. It was something far stranger, a living thing assembled from thousands of smaller living things, each one born from a single fertilized egg, each one cloned, specialized, and locked together into one enormous coordinated whole. Biologists call these creatures siphonophores. They drift through the deep ocean as curtains and threads and spirals, hunting with stinging tentacles, moving without a brain, growing to lengths that defy every rule we thought we understood about size in the animal kingdom. To measure one is to confront a genuine puzzle. Is this a single organism? Or is it a colony, a floating community pretending to be one beast? The answer forces us to rethink what an animal even is. And to begin, we have to clear away the most common mistake of all, the assumption that this is simply a very large jellyfish. The instinct is understandable. See a translucent, gelatinous ribbon trailing stinging tentacles through cold water, and the mind reaches for a familiar word: jellyfish. But a siphonophore is not a jellyfish, and the distinction matters. Both belong to the phylum Cnidaria, the ancient group that also includes corals, sea anemones, and hydroids. Cnidarians share one defining weapon, the cnidocyte, a specialized stinging cell that fires a microscopic harpoon on contact. This is the tool of the group, the family signature. Within Cnidaria, true jellyfish belong to the class Scyphozoa. A scyphozoan jellyfish is a single animal, a bell of pulsing tissue with trailing tentacles beneath, one organism moving as one body. Siphonophores sit elsewhere, in the class Hydrozoa, alongside the smaller hydroids in the notorious Portuguese man-O war. And it is the man-O war that offers the clearest clue. Sailors have long treated it as a single jellyfish, a blue balloon with a venomous sting. In truth, it is not one animal, but a floating colony, a cluster of separate cooperating units disguised as a single creature. The siphonophore takes that same principle and stretches it to an astonishing extreme. Where a jellyfish grows as one integrated body from a single embryo, a siphonophore grows as a chain, budding off unit after unit, each remaining physically attached, each keeping its own specialized job. Imagine the difference between a single sculpted statue and a train assembled from thousands of purpose-built carriages. The jellyfish is the statue. The siphonophore is the train, kilometers long, every carriage genetically identical, every carriage doing a different task. This is why the Ningaloo giant could outmeasure a blue whale without breaking any biological rule. It is not one enormous body straining against the limits of size. It is a colony that simply keeps adding members, extending itself carriage by carriage into the dark. So the record holder is not a jellyfish, and it is not quite a single individual either. It is something in between, a mystery of biology that starts with one fertilized egg and ends with a coordinated society. To understand it, we have to look at how that one egg becomes thousands of bodies. Every siphonophore begins the same way any animal does, with a single fertilized egg, one cell, carrying one complete set of genes. From that cell grows a small larva, and here the story departs from everything familiar. Instead of developing into a single body with organs, the larva begins to bud. It produces a copy of a body unit, then another, each one attached to the last. These units are called zooids, and every zooid in the entire colony is genetically identical to the first, a perfect clone descended from that single egg. This is the heart of the mystery. A blue whale is one genome expressed in one body. A siphonophore is one genome expressed in thousands of separate bodies, all wired together. Picture a stem, a long central stalk, sometimes called the stem or stolon, running the full length of the animal. Along this stem, the zooids sprout in ordered zones, like fruit growing along a vine. In a 40-meter giant, there may be thousands of them, arranged in precise repeating patterns. Some zooids look like tiny bells. Some look like mouths on stalks. Some look like drifting tentacles. Yet none of them ever detaches to live on its own. They cannot. Because each zooid, though it is technically a complete body derived from the ancestral form, has surrendered its independence. Over evolutionary time, these units specialized, each abandoning most functions to master one. The result is a creature that blurs the line between an individual and a group. Ask how many animals are hanging in the water, and there is no clean answer. One egg, one genome, one coordinated whole that argues for a single individual. Thousands of distinct budded bodies, that argues for a colony. Biologists have wrestled with this for over a century, and the siphonophore refuses to settle the argument. It is a body made of bodies, a self-assembled from clones. But identical genes do not mean identical roles. As we move along that stem, from the swimming zone near the front to the feeding zone behind, we find a division of labor so strict that no single zooid could survive the separation. Travel along the stem of a siphonophore, and it reads like a factory line, each station built for one purpose. Near the front sits the swimming section, often called the nectosome. Here, specialized zooids named nectophores form a cluster of muscular bells. They cannot feed, they cannot reproduce. They do one thing, they pulse, jetting water to propel the entire colony forward. Working in coordinated sequence, these bells can drive a creature dozens of meters long through the water, and even steer it, contracting on one side to turn. Some species also carry a gas-filled float at the very tip, the pneumatophore, a single zooid acting as a buoyancy tank. Behind the swimmers stretches the feeding region, the siphosome, and here the diversity explodes. There are gastrozooids, the feeding zooids, each one essentially a mouth and a stomach on a stalk, trailing a long fishing tentacle. There are the tentacles, branching side threads, armed with dense batteries of stinging cells. There are gonozoids, dedicated entirely to reproduction, producing the eggs and sperm that will one day start a new colony. And there are dactylozoids or palpons, defensive and sensory units, some bristling with kinetocytes to protect the whole. Consider what this means. The swimming bell has no mouth, so it cannot eat. The feeding stomach has no means of propulsion, so it cannot move itself. The reproductive zooid does nothing but make gametes. Not one of these units carries the full toolkit of a living animal. Separate any single zooid from the colony and it dies, unable to feed, move, or defend itself. This is specialization taken to a lethal extreme. In most animals, organs depend on one another inside a single body. In a siphonophore, entire bodies depend on one another inside a single colony. The zooids are less like organs, and more like citizens of a society so tightly bound that exile equals death. And that raises the obvious question. What is all this machinery for? Why assemble thousands of specialized units into a drifting chain 40 meters long? The answer hangs beneath the feeding zone, waiting in the dark, a vast and patient trap woven from stinging thread. Below the feeding region, a siphonophore deploys its weapon. From each gastrozoid trails a tentacle, and from every tentacle branch dozens of fine side threads called tentilla. Multiply that across thousands of feeding zoids along a chain 40 meters long, and the result is a curtain, a nearly invisible net of stinging thread hanging in the darkness. Some species arrange themselves into vertical sheets, others into spreading tangles, but the principle is the same. The colony spreads its tentacles wide and waits, drifting almost motionless, turning the surrounding water into a minefield. Each tentillum is a masterpiece of engineering. Packed along it are batteries of conidocytes, the stinging cells shared by all conidarians. On contact, each cell fires in less than a millisecond, driving a microscopic barbed harpoon into the victim and injecting venom. A single brush against the curtain triggers a cascade of firings. In many deep-sea siphonophores, the tentilla are also coiled and colored, and some twitch or glow to mimic small prey, luring fish and crustaceans close enough to touch. When a target strikes the net, the venom paralyzes it almost instantly. The tentacle contracts, hauling the stunned prey upward toward the nearest gastrozoid. That feeding mouth engulfs it, secreting digestive enzymes, breaking the meal down in the stomach shared, through the connecting stem with the entire colony. Nutrients flow along that central canal, feeding zoids meters away that never touch the prey at all. The menu is broad. Siphonophores capture small fish, squid, shrimp, and copepods, and some specialize in eating other gelatinous animals, drifting jellies and comb jellies snatched from the water. In the food-poor deep ocean, this curtain strategy is brilliantly efficient. Instead of chasing scarce prey, the colony maximizes its surface area, becoming a living trap tens of meters across that anything unlucky enough to swim through must pay for. It is a hunting method no single animal could achieve, only a coordinated society of thousands. Yet this raises the deepest puzzle of all. To fire tentacles, contract muscles, haul prey and share food across such distances requires exquisite timing. And a siphonophore does all of it with no brain whatsoever. Imagine a body 40 meters long, stretched through absolute blackness, with no head to lead it and no brain to command it. A siphonophore has neither, and yet its thousands of zoids swim in the same direction, contract at the same instant, and haul in prey with the precision of a single hunter. How does a creature with no central control behave as one? The answer lies in a structure older than brains themselves, the nerve net. Instead of a concentrated cluster of neurons, conidarians spread their nervous tissue across the entire body as a diffuse web. In a siphonophore, this net runs the full length of the stem, threading through every swimming bell and feeding unit. When one zooid detects a signal, an electrical pulse ripples outward, cell to cell, coordinating action without any headquarters. Consider swimming. Along the upper section sits a column of nectophores, the jet propulsion bells. To move, they must beat together, expelling water in synchronized bursts. Researchers studying species like Nanomia bijuga found that a small group of pacemaker zooids near the front fire rhythmic signals down the colony, acting like a distributed metronome. The result is coordinated propulsion, escape jets, even reverse maneuvers, all without a controlling mind. Chemical signaling adds a second layer. When a tentacle snags a fish, chemicals released at the capture site trigger neighboring feeding zooids to orient toward the prey, unfurling their mouths in response. Information travels not through thought, but through concentration gradients and cascading nerve pulses, a chemistry of teamwork. This is intelligence of a strange kind, distributed across a living network, rather than housed in one organ. No zooid understands the whole. Each responds only to its immediate neighbors and the chemistry around it. Yet from these local rules emerges behavior that looks unmistakably purposeful. Hunting, fleeing, feeding, as a unified being. It is a preview of how complex order can arise without a commander, biology's own experiment in decentralized control. But coordination is only half the story. In the perpetual night of the deep sea, a hunter without a brain, still needs to attract its prey, and for that, some siphonophores wield a weapon made of light. Descend past 200 meters, and sunlight vanishes entirely. Below lies the midnight zone, a cold, crushing world extending down thousands of meters, where the only light is the light that living things make themselves. Here, in perpetual darkness, siphonophores have turned illumination into a hunting tool. Bioluminescence is the production of light through chemistry, typically a molecule called luciferin, reacting with oxygen and an enzyme, luciferin. The reaction releases energy almost entirely as light, not heat, an efficiency no human bulb can match. Across the deep sea, an estimated 75% of animals glow, and siphonophores are among the most inventive users of this cold fire. The genus Arena offers a spectacular example. Discovered living at depths near 1,600 meters, these siphonophores produce red bioluminescence, a rarity, since most deep-sea light glows blue. Along their tentacles hang stinging batteries tipped with tiny structures that flicker and twitch. Researchers propose these act as glowing lures, mimicking the wriggling motion of small copepods or the flash of tiny prey. Fish, drawn toward what looks like a meal, swim straight into the lethal tentacles. In a lightless world, the predator becomes a false beacon of food. Why red? Most deep-sea fish cannot see red light, having lost the visual pigments to detect it. A red lure may allow Arena to signal in a private channel, attracting prey that never perceives the trap as unnatural. It is a remarkable coincidence of evolution, or a deliberate exploitation of the sensory blind spots of its victims. Light serves other roles too, startling predators, or momentarily confusing an attacker with a sudden flash before the colony jets away. Each glowing pulse is a message written in chemistry, sent through water that has never known the sun. These living lights reveal how much of the ocean remained hidden until recently. For centuries, the deep was a rumor, glimpsed only in fragments hauled up in nets, torn and unrecognizable. To truly understand siphonophores, scientists needed to descend into the dark themselves, or send machines in their place. And when they did, they found giants no one had ever imagined. For most of human history, siphonophores were nearly impossible to study. Nets dragged from research ships would tear their fragile bodies to shreds, reducing 40 meters of coordinated life into a slick of unidentifiable jelly. To science, the deep-sea colony existed mostly as a mystery in fragments. The breakthrough came with technology, remotely operated vehicles, or ROVs, and manned submersibles capable of filming these animals intact in their own environment. Institutions like the Monterey Bay Aquarium Research Institute pioneered gentle observation, hovering cameras beside siphonophores without disturbing them, documenting behaviors no torn specimen could ever reveal. Then, in April 2020, came a moment that stunned even veteran researchers. During an expedition aboard the vessel Falkor, operated by the Schmidt Ocean Institute, scientists were exploring the Ningaloo Canyons off Western Australia. At a depth of around 630 meters, their ROV drifted into an enormous spiral, a siphonophore of the genus Apolemia, arranged in a vast coiled galaxy of glowing units. The team estimated the outer ring alone measured roughly 47 meters in circumference. By some calculations, the animal itself may have stretched around 45 meters, potentially the longest creature ever recorded. It hung in the water like a colossal string of living beads, coiled deliberately, perhaps to cast the widest possible curtain of tentacles to catch drifting prey. The footage traveled the world, a reminder that the deep ocean still hides giants beyond our imagination. These expeditions do more than break records. Using high-definition video, laser measurement, and careful DNA sampling, researchers slowly assemble the biology of these creatures, how their zooids develop, how they swim, how they hunt. Every dive fills gaps in a story most of humanity has never heard, and each discovery deepens a strange, nagging question. We keep calling the apolemia a creature, an animal, a giant, singular words for something built from thousands of separate cloned bodies. Is that accurate? When we point at 40 meters of coordinated life and say one animal, are we describing a single organism or an entire living community masquerading as one? The answer challenges the very meaning of individuality itself. So what, exactly, is a siphonophore? We have watched it swim, hunt, glow, and stretch, longer than a blue whale. But every time we call it a creature, we brush against a paradox that biology cannot easily resolve. On one hand, a siphonophore looks like many organisms. Each zooid begins as a bud, genetically identical to its neighbors, and each is a modified version of a body plan that could, in other cnidarians, live independently as a polyp or a medusa. In principle, some of these units resemble entire free-living animals. A siphonophore is, in a literal sense, a colony, thousands of individuals fused into a single structure. On the other hand, no zooid can survive alone. The swimmers cannot eat, the feeders cannot move, the defenders cannot reproduce. Strip one unit from the whole, and it dies, useless in isolation. They share a common gut, a common nervous net, a common fate. By that measure, they are not individuals at all, but organs, no more separate than your heart is separate from your lungs. This is where the question turns philosophical. What makes something one individual? Is it shared genes? Then identical twins would be a single person. Is it a single body? Then what of the fused inseparable zooids? Is it the inability to survive alone? Then the cells of your own body, none of which can live independently, are the true individuals, and you are merely their colony. The siphonophore holds a mirror to ourselves. You are trillions of cells, genetically identical, specialized into tissues, coordinated without any single cell in command. From a certain angle, you too are a colony that has forgotten it was ever many. The siphonophore has simply drawn that boundary in a different place, leaving its units visible, still faintly separate, cooperating in the open dark. Perhaps the question one or many was always the wrong question. Perhaps individuality is not a fact of nature, but a line we draw for convenience. In the black water of the deep, the siphonophore drifts on, indifferent to our categories, embodying a truth that dissolves them entirely. Many bodies, one impossible life.